Okay, tonight I'm going to start with the digestive system, and then I'm going to cover the metabolism part of this lecture. We're going to have a break after the digestive system so that you can get more food to digest. Then I'll cover the metabolism lecture, which usually takes me about an hour, 45 minutes to an hour. And I will also finish the autonomic nervous system. I didn't get through all the slides last Thursday before I started losing my voice. Someone needs to make these winds stop, please. It's like a dust bowl out there. And then I have some goodies for you. Well, a goodie for you. And I suggest that you hang around and work on it. And I also suggest that on Thursday, when I'm done telling you about skeletal muscle contraction, you also hang around and work on it. Okay? Right. So the digestive system, I affectionately call it the poop chute. I do. It has another name, of course, the alimentary canal. And I'm going to explain that to you in just a moment, what that means. The alimentary canal is basically a continuous tube that starts from your oral cavity all the way down out through your anus. It is a continuous tube that is actually still exposed, if you will, to the outside world. It starts from the outside, it finishes on the outside, right? If we're talking about the digestive process, the first thing we need to do is ingest the food, which is what many of you are doing right now. So you put the food in your oral cavity and you chew it. That is a type of mechanical digestion. But chewing is not the only place where we're going to see mechanical digestion. We also have mechanical digestion in the stomach and the small intestine, for example. I will address those in just a moment. Propulsion means you need to have this bolus of food, and let's use that terminology. When you swallow and you feel that food going down your esophagus, it is called a bolus, B-O-L-U-S, of food, a bolus. When it gets to the stomach and all the secretions are mixing with your food, then we call that mixture chyme, C-H-Y-M-E, chyme. And it's chyme from the stomach through the small intestine until it passes through the ileocecal valve. Don't worry, I'm going to show you pictures of these things. We're going to go through the alimentary canal step by step. We will address what secretions are released, what digestive enzymes are released. We will also address which types of mechanical digestion occurs and where. Then we're also going to talk about absorption. How do we absorb our nutrients? What do they have to look like? Can they be polymers, large molecules? No. Our cells don't have the transport for large polymers. I'll remind you, we started this entire lecture unit with membrane transport for a reason. Everything we are studying for this entire unit hinges on membrane transport. If I had to pick one theme, that would be it. Down to neuron physiology, digestive system physiology, problems with transport like diabetes mellitus, which I will address, membrane transport. So how do we absorb these nutrients? The last part of this lecture for metabolism will be a very brief introduction of what happens. How do we burn these things? What do we do with them? What do we do with amino acids? What do we do with carbohydrates? What do we do with fatty acids? What do we do with the nucleic acids? And then lastly, whatever we don't absorb, of course, we're going to defecate. Okay, so now I'm going to get, I don't mean to be crude and crass, I'm just trying to be fun and give you a visual of this alimentary canal, okay? When my boys were younger, they would wake up, they have 4.5 years between them. But my oldest, his favorite for breakfast, I would ask him, what do you want for breakfast? His answer was always peas and corn, peas and corn, peas and corn. That's what he wanted. So I would get frozen peas and corn and microwave it and say, here you go. And he was so happy. I'm like, don't you want Cocoa Puffs? Don't you want, can you be a normal child? Do you have to be that healthy? Peas and corn, Mommy, peas and corn. And then when his younger brother was old enough to eat table food, both of them, peas and corn. But at that point, the oldest one could actually say peas and corn. But when I was changing their diapers, it was, and I'm an exhausted parent, right? Very little sleep. I'm deprived. I hallucinate sometimes when I'm so tired. And you all know that I start to have parties in my head to entertain myself. I've shared that. Okay. So I would change their diapers, and little children do not masticate. That's mechanical digestion very thoroughly, especially when it's little finger foods like peas or corn. And I'll remind you that both of those vegetables actually are surrounded by fiber, cellulose. And that is a type of carbohydrate we cannot digest. If you do PhysioX number 8, activities 1 through 4, it will reinforce this. So I changed their diapers. They've pooped, they've defecated, and I opened the diaper, and in their poop are full corn kernels and peas, like intact, because they didn't chew, they just swallowed it. It's wet, it's small, they just swallowed it. So because I'm like, I can't handle this right now. These children barely slept last night. And the party in my head was, they're totally intact. I think I could probably pick them out, wash them, and they'd be good to go. I didn't do it. It's just the part. Hey, when your youngest wakes up at exactly 10 minutes to 5, I could put that child to bed at 4:45 in the morning, and 5 minutes later, good morning, Mommy. It's a good day. I'm like, oh, I can't. I didn't do it. But I want you to have that visual. Let's say I have a giant piece of corn, and I'm going to tie around it dental floss, okay? I secure it. It's not just a loop. I'm like all directions. Long piece of dental floss, long piece. I put that corn in my oral cavity. I swallow. It enters my oral pharynx, my oropharynx. Travels for a short time into my laryngopharynx. Meanwhile, the dental floss is going with it. Then it enters my esophagus. It comes down to my stomach. Dental floss is going with it. It then goes into my small intestine. Down Jones Industrial, duodenum, jejunum, ileum. Did you understand what I just did? Down Jones Industrial, duodenum. The duodenum is called that because it is roughly 12 fingers. That's what it means, 12 fingers in length. Then we have the jejunum, which is a site of major absorption of nutrients. And then the ileum, still a site of absorption. So it goes through all of my small intestine. Then that piece of corn goes through my ileocecal valve. It goes up my ascending colon. That's part of your large intestine. It goes across my transverse colon, still a part of the large intestine. It goes down my descending colon, still part of the large intestine. And then it travels through my sigmoid colon, which is called that because it is S-shaped. Then it enters my rectum. And then finally I feel that pressure. We're going to talk about defecation. And I defecate it out. And now I have a piece of dental floss that I can go a continuous tube. Come on, it would be fun if we could actually do that. You can't unsee that in your head now, can you? Oh, I'm sorry. Strap in, it's just going to get worse today, okay? It's just going to get worse. So let's talk about the types of mechanical movement, propulsion. Swallowing, deglutition. The esophagus, we have peristalsis. I'm going to talk to you about receptive relaxation and how you have to be careful with that. This is where our lecture is not even rated PG, okay? It's not. And you're going to be like, no, you just wait. Peristalsis, have you seen a snake eat a rat or a mouse? And you can see the bulge, and it pushes, it constricts. The muscles constrict behind the bolus, but ahead of it, the muscles relax. That's called receptive relaxation. And that makes it easier for that rat, that mouse, or that bolus of food that you're eating to move through the alimentary canal. So receptive relaxation occurs ahead. The muscles relax so that it's easier to squeeze that bolus of food further down the alimentary canal. The stomach also has unique mechanical digestion, most notably retropulsion. After you eat, I'm sure you've heard a lot of the noises from your stomach. They can get quite loud, can't they? That's from the churning. That's what retropulsion means. Churning, mixing, churning, mixing. And there's a lot of opportunity for mixing because in the stomach, we have macroscopic folds called rugae. And I will show you what those look like in just a moment. So it makes all these squeaks and bubbling sounds, doesn't it? And you're like, I haven't eaten, and my stomach is growling. No, you ate. This is called retropulsion, and it can get very loud at times. You put your ear to someone's stomach if you're laying on their stomach, and you're like, whoa. It's even louder when I do that. And then we get into the small intestine. We still have peristalsis, but we also have segmentation. And segmentation reminds me of a cat. You know how they start doing this? A lot of people describe it as they're making muffins. Okay, that's kneading the dough, if you ask me. But they do this. Segmentation. Localized contractions to further mechanically break down the bolus of food, the chyme. We can also have pendular motion. What does that look like? Well, peristalsis can work in reverse, and then forward again, and then reverse. So it's like a pendulum, swing back and forth. More mixing means more opportunity for the digestive enzymes to do their job, which means then you can properly absorb those nutrients through your membrane transport mechanisms. If you go back a little bit, if you eat a meal, like if you're eating a lot of food that I brought you, making a meal out of it, by the time the stomach adds all of its secretions, that meal can have a volume of 1.5 liters once it's liquefied. And you know what I mean by liquefaction, right? You've thrown up before, I'm sure, and out comes your meal. Okay, that will take about 3 hours for it to completely leave your stomach and enter the small intestine. And the reason why we don't just go bleh from the stomach, all of that, into the small intestine is because there's a lot of acid and it needs to be neutralized. I'm going to talk about that as well. Then we get to the large intestine. And the large intestine still has this peristaltic action, moving the feces from one part to the next. But we call this mass movements because you have this big amount of contraction in your large intestine about 1 to 3 times a day. And it pushes the feces down through your sigmoid colon into your rectum. Now, when it gets to your rectum, it only needs about 25% full, and you get the urge to defecate. The question is, do you obey that signal, that biology signal from your body? Do you? Some people pinch it back, and they don't go. And the large intestine, if it could talk to you, would say, fine, maybe now is not a good time, but you are going to listen to me eventually. And then later in the day, you get another one. And it's usually, we have this rule of the gut, food in, feces out. Because that receptive relaxation ripples through the entire alimentary canal. The entire alimentary canal. So 1 to 3 times a day. I hope you all are defecating at least 1 time a day. Are you? In my household, our dinner conversation was quantity, color, and consistency. That is what we talked about of our feces. Yes, I was the only female, not counting the female dog. I was outnumbered. If you can't beat them, you join them. Like, I already went 3 times before it was 9 o'clock in the morning. So we would have competitions. I know, you're laughing, but hey. My nutrition professor from college, UCSD, Dr. Saltman, he would say to you, you need, and excuse my language, these are his words, not mine, you need to take a shit every day. And his research, he would go to Africa and study the natives there, and he would actually measure their piles, and again, of shit. He would measure them. And he noticed that the more times they defecated and the bigger the pile, they had very little incidence of colon cancer. So he would say, you need to make sure you need to defecate every day, at least once. Now, the irony is not lost on me. Sadly, he died from colon cancer. I know. He was very entertaining. I adored that man. Okay, so food in, food out. Traditionally, if you eat something within about 20 minutes, that receptive relaxation will hit all the way down to your sigmoid colon and your rectum, and you will have the urge to defecate. This is called or referred to in some books as the law of the gut. Food in, poo out. Many adults don't obey those biosignals from their own body. They don't. Oh, I can only defecate in my bathroom. I can't defecate on when I'm in school on campus. I just can't. I can't do that. I can't. Okay, so you pinch it back. That's what you call it. You do that often enough, and you can become anal retentive. That is a real thing. There are two groups of people that have a high incidence of being anal retentive. That's toddlers and teenagers. And the reason is, for toddlers, they know at that point that it's a private thing to do. And if they're in preschool or daycare, usually they have stalls, but the stalls oftentimes are open so that the teacher's assistant or the teacher, whoever is there, parent helper, can check and help those little kiddos wipe. And we do want to help them because they're like T-Rexes. They've got these, and they can't really reach. They can't. They just kind of smear it up their backside. So they're basically pooing for everyone to see. And they don't feel comfortable with that. I don't blame them. Teenagers, why do they become anal retentive? They spend the majority of their day with their peers, with other teenagers. I remember my son, and both my sons were masters at defecating. They for sure were. But the bathroom doors in the men's bathroom would not lock. They were busted. My son actually called me, said, "Mom, I need to just come pick me up from school. I need to go home. I have to go to the bathroom." And I said, "H, you can do this at school." "Mom, the locks are busted. And the guys, they think it's funny to kick open the door and catch you doing your business." I was like, that cannot happen. So I went there, picked him up, took him home, brought him right back, and went to the desk, the front desk, and said, "This cannot happen. You need to make sure those locks work." So they did. They fixed them. But those are the two groups that can be anal retentive. Excuse me. Sometimes they get so anal retentive that the large intestine, if it could talk to you, would say, "Forget it. You keep pinching it back. You keep overriding my biosignals that I'm giving you. Fine. I'm just going to be a lazy gut." And then the large intestine, I'm going to remind you about, it's really good at absorbing more water. And that means that fecal material gets more and more desiccated. It doesn't have a good mucus layer around it, so it can just slide right out of you. And then it becomes a fecalith, which means fecal stone. And it will be very difficult to pass. And if you are the nurse, I suggest you double glove, because you are going in there like Nemo, as Chris Hardwick would say, and you are going to do a digital extraction on that patient. Maybe now is the time to reconsider your career goals if you don't want to do that. It's gonna happen. Now let's talk about something else. This receptive relaxation, this law of the gut, is supposed to be food in, feces out. Okay? But it can work in reverse. It's going to happen. For some of you getting a visual, it can happen in reverse. I encourage you to look up Chris Hardwick, who is a comedian, on YouTube, and look up his Nurses sketch. That's what it's called, Nurses. He does the best thing to explain this law of the gut and how it can reverse its direction. Receptive relaxation going back up through the alimentary canal. Now, some people, I don't mean to be crude, this is a real thing. People go to the hospital for these issues, okay? You cannot laugh. But there is a certain point if you insert something in your anus, as Chris Hardwick would say, it becomes very greedy. And it's almost like the seagulls in Finding Nemo. Mine, mine, mine. Receptive relaxation works in reverse, and whatever you're putting back there can be pulled up more superiorly through I know. Now, we don't have to be crude and talk about different types of toys to do that with. But here's my cautionary tale. If you are using an anal thermometer and an infant, or anyone for that matter, you don't just stick it in there and then turn your back doing something. You better make sure you're holding on to that, because again, mine, and you turn around and you're like, where, you're going in for that. Nemo, you're going in. Digital extraction all over again. Yes, you are. Hold on to it. I'm just saying, if you're into that sort of thing, make sure. I mean, they even have the toys where it's on a string. You can pull. You can. You're laughing. It's true. It's true. True stories. What have my previous students told me had to be extracted from someone's anus? True. Goldfish. And I'm not talking about the little crackers, a real goldfish. True, a light bulb. And even Chris Hardwick in his sketch says, what do you say when you pull a light bulb out of someone's anus? And he does this, I have an idea. He goes on to say that a nurse, and one of the nurses just shouts out, and he says in his sketch, I love to ask nurses what's the best thing they've pulled out of someone's anus. And right away a nurse in the audience says, a broomstick. And he's like, a broomstick. And he says, do you hear how calm and measured her voice is? Like, this happens all day long. One of my students, a patient, came in with a mag light. You know, the big flashlights? Okay, you got to go in there to get that. And nurses are hilarious too, especially the ones in the emergency room. They will start communicating, like there's a secret phrase, which essentially means you need to come to the emergency room if you're on a break and see this. You're not going to believe this. And you're like, okay. So a mag light. And then Chris Hardwick also says he saw this tough biker dude, like Sons of Anarchy. And he was a nurse. And Chris Hardwick, being a comedian, he said, pal, what's the best thing you've ever pulled out of someone's anus? And the nurse answered, my fist. I can't do that sketch justice, but it's awesome. Now I'm going to have to listen to it again on my way back to the hotel. I'm just saying you need to be careful with it. Okay? If that's your fetish, fine. That's your fetish. Good for you. But be careful. Receptive relaxation works in reverse. Mine. Good. Can you remember that? That was a public safety announcement. You're welcome. Yes, I am posting this on YouTube. I did give proper credit to Chris Hardwick, just so we know. Chris, I'm not stealing your material. You just inspire me. Up on the screen, you are seeing different parts of your alimentary canal, and you are seeing what secretions are released from each place. I have color coordinated them, even on the later slides. So in yellow is an enzyme that can digest lipids. It's called a lipase or a cholesterol esterase. Digests lipids. In theory, fats are digested beginning in your oral cavity. Yeah, but in PhysioX number 8 does address that, but the activity is negligible. It's negligible. I put it there, but we're going to just agree that it doesn't, why? Why? Its activity is not robust. In kind of the salmon reddish color, I have enzymes that are going to digest carbohydrates, polymers, into smaller sugars, even down to glucose. Then I have in green enzymes that are going to digest proteins into smaller peptides or even single amino acids. And then I have some special considerations. In the bright blue, I am listing nucleases. A nuclease is an enzyme that breaks down nucleic acids. I know it probably you didn't think about it, but when you're eating an apple or a banana or any of the food that I'm bringing you, like the grapes, there's DNA material in there and RNA. And you can use those. You can use the nucleotides for your own purposes to replicate DNA or make RNA. It doesn't go to waste. Then I also have up here in gray a secretion from the stomach called intrinsic factor. This is not the first time you've heard about intrinsic factor. You learned about it in lecture unit 1 when you learned about your anemias and you learned about vitamin B12 deficiency. In order for the ileum to absorb vitamin B12, it must have intrinsic factor. We talked about that, remember? Now up at the top in the intestinal mucosa, I have in blue, like a gray blue with green border around it, enterokinase. Let me explain to you what that does. Enterokinase is an enzyme that will activate these 3 enzymes. Those 3 enzymes are produced in the pancreas, and they are made in a precursor form, a zymogen. This is not the first time you've heard me tell you about a zymogen. You remember all your clotting factors? Fibrinogen, right? So those were all proteins that had to be activated. Enterokinase will activate trypsinogen into trypsin, chymotrypsinogen into chymotrypsin, and procarboxypeptidase into carboxypeptidase. You don't want them activated until they are in your small intestine. If they get activated while they are in your pancreas, understand they digest proteins. Understand proteins are made inside cells through gene expression. Understand your pancreatic tissue will start to be digested. And you will know exactly where you are and what time of day when you started to feel that pain. And you now have pancreatitis. For sure, you're going to be going to the hospital, And it takes a long time to get those 3 little stinkers under control. Expect to be there about 10 to 14 days, depending on the... Yes, it is not something you want. Most of the time, acute pancreatitis is from a blockage in the pancreatic duct, or where the pancreatic duct meets with the bile duct from a gallstone. Then all those enzymes dam up and they can auto-catalyze themselves. They can convert themselves. There's a story behind that, why that happens, but I'm trying to get through the material tonight to give you your goodies. If it is chronic pancreatitis, if you have pancreatitis 3 times in a year, that is chronic, and you are not long for this world. You are not. And that is usually caused from alcoholism. It is. Again, very, very painful. Question, which one would you rather have? Pancreatic cancer or pancreatitis when it comes to pain? Pancreatic cancer. I mean, you're not going to live long either, but it's not as painful as you digesting your own tissue. Yeah. Okay, next slide. So let me summarize this. If you look carefully, we start digesting carbohydrates in our mouth. If you chew any of the crackers or the chips that I have up here long enough, masticate them, get them all liquefied, it starts to taste very sweet. And it's because that amylase is breaking them down into simple sugars. If you look at the stomach with pepsin, which is made as pepsinogen, it is the first place where we have protein digestion. And something else that PhysioX is going to show you repeatedly in these 4 activities is that enzymes are substrate specific. That means an amylase cannot digest lipids. A lipase cannot digest proteins. They are specific. Number 2, they have an optimal pH. An optimal pH. Pepsin is a unique enzyme that actually has optimal activity in a very acidic environment. Well, that's good because it's the hydrochloric acid that activates pepsinogen into pepsin. In your PhysioX, you're going to be playing with different enzymes in different pH values, different pH buffers. And you will see that there is peak activity in one of those 3 that you're playing with. Very basic, more neutral, very acidic. Third, enzymes are temperature sensitive. If you boil an enzyme, it's going to lose functionality. But we can freeze proteins. Enzymes are proteins. I used to be able to say that. Now we know of some RNases that are enzymes and they're not proteins, but most enzymes are proteins. They're temperature sensitive. You buy a whole bunch of steak and chicken at Costco. You want to preserve them. You wrap them up individually, and where do you put them? In the freezer. Freezing will stall bacterial growth, but if you take those things out, can bacteria start growing again? Absolutely. You'll notice in the PhysioX, when we freeze an enzyme first, it can still work once we bring it back up to normal body temperature. It will. The 4th thing, and PhysioX does not address this, but enzymes are sensitive to salt levels. They are. And if you think about the beef sticks that I bring you, beef jerky before there was ice, before there were freezers, on ships, the sailors would bring meat that was dehydrated and salted, right, to kill off the bacteria and their enzymes. So that is something PhysioX does not address. Protein digestion, lipid digestion, digestion of carbohydrates and nucleases continue in the duodenum, the small intestine, not just from enzymes from the pancreas, but also from the intestinal mucosa, and I'll explain what that is. We're now entering our alimentary canal. We're going to go step by step. If we're talking about the oral cavity, you get a lot of secretions from your salivary glands. You have 3 of them. The largest of the 3 are the parotid glands. Then you have submandibular glands. And then you have sublingual glands. One of my previous dentists loved to press right underneath my tongue and get my sublingual gland to squirt its secretions at the dental hygienist. I didn't do that, but he would do that. I don't know if he had the hots for her. I'm not really sure, but I didn't appreciate that. I'm like, that's just rude. Just tell her you like her. Why do you have to act like you're in 6th grade and like pick? I don't know. It was just weird. But have you ever accidentally, with your tongue pressed on your sublingual gland and all of a sudden water starts shooting out? Most people call that, oh, I just gleeked, right? That's you putting pressure on your sublingual gland. Through these salivary glands, you make about 800 milliliters to a liter of saliva a day. And if you look at the disposable water bottles throughout the room, that's about as much as you're making. Well, that's 500 milliliters. Now think about 2 of them. Yeah, so if you went around with those bottles, the bottle, it's about a liter a day. If you don't produce enough saliva, and there are some medications that can prevent that some people might take, you will have a higher incidence of cavities. You will. Because in the saliva, not only is there the salivary amylase, lingual lipase, negligible, but there's also bactericides. There are chemicals there to fight off the bacteria that are living in between your gums and your teeth. So it's important to make saliva. What else can I say? There's also antibodies that are made. There are enzymes that are there to break down bacteria that are in your mouth, absolutely. And salivary glands can be more serous, S-E-R-O-U-S, which means they're producing more liquid-like. That would be the parotid gland. They can be more mucus producing, like the sublingual. Ever notice that your dental, I love getting hand scaled. Do you know what that is when you go to the dentist? Do most of you use the Cavitron, or the hygienist uses that machine that goes to clean your teeth and break up your plaque? I can't do that. I can't. I'm like a cat attached to the ceiling. I can't handle the noise. So I make my poor hygienists hand scale me. That's where they get the pick and they scrape and they scrape. I love it. I just lay there. I sound like I'm in the wrong place because this is what you hear down the hall. And she just I call it my mouth massage. That's what I love, going to the dental hygienist. Love it. I'd go every week if I could. Do you not like going to your hygienist? Dentist? I love it. Love it. It's the best thing that my insurance pays for. So right here, if you ever get hand scaled, your lower four incisors, your lower four teeth here, they get impacted with a lot of plaque because of that sublingual gland making a lot of mucus. It's like a glue, and all the bacteria get there and some food. Even if you floss, it still is there, and they'll spend a lot of time scraping that off. The submandibular glands are a mixed gland. They are about equally serous, liquid, and mucus. Moving on. After we swallow, that bolus of food enters your esophagus, thank you peristalsis, and eventually it enters the stomach. Now let's take some time to address the stomach. It is a short-term storage facility. It's going to take some time to churn the food around, retropulsion. It's going to take some time for mixing of the pepsin with the food that you just ate for protein digestion. Pepsin, again, is made as a precursor, a zymogen, called pepsinogen. The cells that release pepsinogen are called chief cells. What activates pepsinogen into pepsin is hydrochloric acid. Parietal cells release hydrochloric acid and intrinsic factor. We, again, don't want an enzyme that digests proteins to be active inside the cell. You will start to digest yourself. You don't want that to happen. We also have cells in the stomach that produce a lot of mucus, bicarbonate-rich mucus. And this ends up being a very good protective barrier, which I will talk about in more detail when we get to my ulcer discussion. So not only do we have mechanical breakdown, but we also have chemical breakdown. As I said, we have liquefaction of the food on a previous slide. Your food, a typical meal, turns into 1.5 liters and takes about 3 hours to actually be expelled from the stomach. Gastrin, I will address again in just a few slides, is a hormone. And gastrin is a big player for getting the parietal cells to release hydrochloric acid. Hold on that thought, and I'm going to tell you about that. In the stomach, we have gastric glands. These crypts, if you will, these invaginations. Towards the top of the gastric pit, gastric gland, we have mucous neck cells, making the bicarbonate-rich mucus. Down at the bottom, these gastric glands, we find enteroendocrine cells. They are single cells that release a hormone. In this case, gastrin. If you're taking notes, gastrin. We also find chief cells. They are called chief cells because they are the most numerous deep in these gastric glands. And then shown here in kind of the salmon color, we have the very large parietal cells, which release hydrochloric acid and intrinsic factor. Now in gastric glands, these 3 cells are found in 80% of the stomach, found in the cardiac region, which means it's close to the heart. It's on the other side of the diaphragm, but it's still close. The fundus is the most domed shaped part of an organ, and the body of the stomach. We also have, in a different location, another type of gland called the pyloric gland. And the pyloric gland, found here, we're going to find that we have the enteroendocrine cells, same picture. That's where we find these enteroendocrine cells that are making gastrin. We still find mucous neck cells, and not so much the parietal cells and chief cells. These two types of cells are the big players in this region of the stomach. That's not to say you can't find them in the gastric glands. It's not to say you can't find them all. I'm just saying, proportionally speaking, in the gastric glands, mucous neck cells, chief cells, and parietal cells. Proportionally speaking, in the pyloric region, that's this region down here next to the pyloric valve that empties the chyme into the duodenum, the majority of the cells are the enteroendocrine cells and mucous neck cells. You've heard of Pavlov's dogs, right? Have you heard of that? Can anyone tell me what happened with Pavlov's dogs? He had them trained so that if they heard a bell, they started salivating because they equated that sound with, hey, food's going to come. And their juices started going, saliva and even the acid in their stomach. And I'm sure you've seen YouTube videos where someone is showing their dog or cat and all they do is open like a bag of chips or something, or open the can of cat food, and all of a sudden, whoop, the animal comes running, don't they? Oh yeah. For me, my dogs, when they hear me eating an apple, or even my parrots, if they see or hear me crunching on an apple, all of them right there, immediately. They even hear it from upstairs. They come running. Well, the parrots don't run. They start squawking in their cage. So that's a Pavlovian response. We behave the same way. When we see food, as in not seafood like crab or lobster, when we see food, that's called the cephalic phase. And that triggers your stomach to start releasing this acid. Then we have a gastric phase. And the gastric phase means food is actually touching your stomach mucosa. Physical contact stimulates the cells, and they start releasing their secretions. And as if that's not enough, then there's an intestinal phase where, again, once the food is contacting the small intestine, that triggers more feedback to the stomach, tells the parietal cells to release more acid, more hydrochloric acid. So 3 phases in time, if you will, chronological: cephalic, gastric, intestinal. But what exactly is the mechanism to get this acid to be released? It is either local, which means the physical contact of the food in your stomach causes the parietal cells to release more hydrochloric acid. We're going to see that enterochromaffin-like cells are responsible for this. Specifically, they are going to release histamine. Shown up here, histamine in the red. Then we're going to see a nervous system component. The vagus nerve is a preganglionic neuron that extends from your brainstem, and it actually is the only cranial nerve that can extend into your abdominal cavity, all the way to the left colic flexure, which means the transverse turns into the descending. The vagus nerve releases acetylcholine, and the postganglionic neuron releases acetylcholine. That was in your quiz, remember? Yep. And then lastly, we see the enteroendocrine cells, the G cells, that are in the stomach, mostly in the pyloric region. They have a huge involvement, in releasing acid. So seeing food, the touching of food in your gastric and intestinal mucosa keeps stimulating this acid release. This is called potentiation. It really is a form of positive feedback. More is leading to more is leading to more. This can be a problem in some people. It can lead to peptic ulcers, which I'm going to address soon. So let me, before you have a break, a short break, tell you about this acid secretion. What actually extrudes the hydrogen ions into the lumen of the stomach, the acid, that will activate pepsinogen into pepsin, is this pump called the hydrogen potassium ATPase. These parietal cells, and that's what this is showing you, have a receptor for acetylcholine. That's the nervous system component. They also have a receptor for gastrin, the hormone that is the big heavy player to get a lot of acid to be secreted. And then we also have a receptor here for our local chemical signal, histamine. And we did learn about our second messenger systems, if you recall. We learned about the big two, IP3, DAG, and calcium. And we also learned about cyclic ANP. If you look inside this parietal cell, two different stimuli feed into the IP3, second messenger system. And second messenger systems are an avalanche effect. You get them going, it gets bigger, it gets bigger, it gets bigger. Now you have two of them feeding into that. It's crazy. We have another one, the local factor, using the cyclic AMP. Doesn't matter. We have 3 different ways to stimulate second messenger systems in the parietal cell, leading to copious amounts of hydrochloric acid. I'm going to tell you about the treatments that we can use for peptic ulcers when you come back from a short break. Please go take a 15-minute break.